Hi everyone. Today I am going to connect three topics that link genes, hypotheses, and disease. Let's begin with APOB. I found gene a polypoprotein B on the list of genes related to cardiovascular disease. Among cardiovascular-related genes, I selected APOB located on chromosome 2, and it plays a key role in lipid metabolism. This gene produces two main protein forms. APOB48, made in the intestines essential for forming chymicrins to transport dietary lipids. APOB100, produced in the liver and required for LDL particles to bind LDL receptors and enter cell. From the lipoprotein structure shown in the PPT, APOB100 sits on the LDL surface like a barcode that allows cells to identify and clear LDL. If APOB100 is defective, LDL cannot be cleared efficiently, leading to elevated cholesterol and accelerated atherosclerosis. Several mutations contribute to this. A well-studied example is an R3500Q mutation associated with familial defective EPOB-negative 100, which reduces binding affinity to LDL receptors and causes premature heart disease. Conversely, certain truncating mutations, such as APOB46 or APOB29, may lead to hypopithelia porphyria, which sometimes results in fat malabsorption or fatty liver. Therapeutic approaches targeting this pathway include statins, mepomorphin, and newer RNA-based therapies such as enzalutamide. The next part is a design experiment about does light exposure at night affect sleep-wake cycles in mice. My hypothesis is light exposure disrupts the sleep-wake cycles in mice. Two mouse groups were studied. Group A, constant light exposure. Group B, standard 12-hour light-dark cycle. For 2 weeks, motion sensors continuously recorded behavior. My expectation is that the control group will show stable day-night activity patterns, while the constant light group will display fragmented or irregular rhythms and potentially decreased activity. This experiment helps model how light pollution affects biological clocks with implications for shift work, mental health, metabolism, and modern artificial lighting environments. The next part is the research paper. The key points of this paper are current biomarkers for CVD have limitations as diagnostic tools. Exosomes have the potential to be more efficient and accurate than traditional biomarkers. We have to overcome some current challenges to the clinical use of exoamoebal biomarkers. Let me introduce these two traditional biomarkers to heart muscle. The first one is troponins. When the heart muscle is injured, such as during a heart attack, troponins leak into the blood, so measuring chopstick levels helps diagnose AMI. The second biomarker is cytokines. Cytokines regulate immune responses and inflammation. They are produced when coronary arteries are blocked, and different types are associated with different cardiovascular conditions. However, both biomarkers have limitations. Recently, researchers identified a new biomarker, exosomes. Exosomes are extracellular vesicles containing proteins, lipids, and RNA. They are released by many cell types and play a role in cell-to-cell communication. Because of this, exosomes may serve as more accurate biomarkers for cardiovascular disease. A key component carried by exosomes is microRNA. MicroRNAs are small, non-coding RNAs that regulate gene expression by binding to mRNA and preventing translation. Their synthesis involves several steps. So, how are exosomes connected to cardiovascular disease? Endothelial cells and macrophages release exosomal microRNAs that influence plaque growth and stability. Why are they reliable biomarkers? Because RNA-based measurements are more sensitive and specific, but challenges remain. In conclusion, from genes to RNA to diagnostic biomarkers, the intersection of molecular biology and clinical medicine is powerful. As research progresses, genetics, RNA therapies, and exosome-based diagnostics may allow earlier detection and more personalized treatment, moving us closer to preventing cardiovascular disease before symptoms appear.